Hello, everyone. Welcome to CDD's Q1 webinar. I'm delighted to be sharing the stage, say that 7 times fast, with Chris Waller and Dimitra Agrafouidis here, who are two of the people who know as much as anyone that I know about what it's like for drug discovery informatics for big pharma, which is the title of the webinar. And I would like to make a short public service announcement before introducing them, which is that if you have any questions, please ask them in the Q&A. And if at the end, either Charlie or Joshua will gather them and we'll ask as many and answer as many as there's time for. So we want to engage your brains, and that shouldn't be hard to do. I want to share a few things about Dimitri's background, initially from Greece. Professor Henry Rzepa is someone that I've heard give a talk at my last company, and he would talk very much about new chemoinformatics technologies. Professor Streitweiser is someone I worked on the 7th floor Latimer Hall at Berkeley for my PhD, which is right like next door to him pretty much. And when I was thinking of which professors to work with, he wanted me to work on putting as many negative charges as close together as possible on a molecule. I wanted to also mention in passing that Christopher Lipinski of the famous Lipinski Rules of Five also worked with Professor Streitweiser, and he wrote this organic textbook, Streitweiser and Hethcock. So there must be something in the coffee there at Strata, Dimitris, that gets all these interesting people. And then the one that I don't know too much about what you did with the Nobel laureate E.J. Corey, I know about his retrosynthesis and forward synthesis work, but is there anything interesting from that time over there at Harvard to share? Just going
Uh,
back in the time machine.
Hi, Barry. First of all, I'm delighted and honored to participate in this webinar. I had enormous respect for you and what you've done with CDD. To answer your question, my project actually was a fantastic place. I mean, I worked on the LASA project,
Oh yeah, that's fun.
25, 30-year-old project. Stands for Logic and Heuristics Applied to Synthetic Analysis, essentially an expert system for designing retrosynthetic routes for complex molecules from readily available starting materials. My specific project was encoding into it. Again, it was a very, very sophisticated system. But encoding into it stereochemical control and preferential reactivity by three-dimensionalizing the molecules
Mm.
as the retrosynthesis was happening inside the computer. So historically, it was based on rule bases, right? Chemical synthesis, organic synthesis, rules. And my project was to introduce 3D into this.
Excellent. That's a perfect transition for me to mention 3D pharmaceuticals. We did not plan that, but I had dinner with Ray, one of the founders of 3D Pharmaceuticals way back when. And the thing that's fascinating to me about that is 3D got acquired by J&J, and you developed this ABCD informatics platform. And as folks know, sometimes when smaller companies get acquired by bigger ones, they might just go for the furthest along drug candidate and squash some of the pipeline or spin that out as a company, but even more so for the enabling technologies. And somehow you did the opposite. You managed to get ABCD to get more funding and develop further. And you presented it to us at CDD way back when. And it was amazing. It was just groundbreaking. It was beyond what I had seen at the time. So I'm kind of curious to hear. Obviously, they promoted you 2 more times there. You're very successful, but you're also successful developing new technology, really cutting edge technology within a much larger company that had been started in a smaller company. So, yeah, anything about how you shepherd it through
Um,
those waters? Because that's just
that's a great.
amazing. Yeah.
First of all, thanks for the kind compliment, Barry. But it's fascinating, actually. I reflect back on one should not underestimate the impact of luck and circumstance. Okay, but there is method to the madness as well. Let me try to highlight those two items here. As I reflect back on my career, I think one thing that I did well, very proud of, and it has helped me tremendously, is that not only did I do relevant work for the companies I worked for, but I also published very extensively. Mm-hmm. And 3DP, you guys would remember, we had tons of publications out there, very good papers. Mm-hmm. So the team was very well known in the industry. And when J&J acquired us, of course, they acquired us primarily for an asset, right, that we had, or a couple of assets that we had in our portfolio, but also because of the talent and the expertise we could bring in. And I happened to run into truly visionary leadership at the R&D side. And also, I have to say, I was very impressed, fortunate, benefited greatly from it, but I was also very impressed by how advanced J&J was in managing acquisitions. People have asked me, how did it feel when you joined that company? And I tell them, most of us flourished. I mean, their careers exploded. The moment the ink dried, the signature dried, every one of us was given the same opportunities and the same behavior, the same sort of, they treated us the same way as the most seasoned veterans. Once we're part of the family, we were part of the family. And were evaluated individually, and that was their brilliance. J&J is a brilliant company in ingesting and incorporating acquired parties into their culture. At least that has been my personal experience.
Yeah, J&J is famous for that. My business mentor was Alejandro Zaffaroni, whose initials Alza is named after, which J&J had bought. I could go deep about all of these. I think the only other one, because we'll talk with Chris about Pfizer. So Novartis, we're lucky to have our own Peter Geddick there, who shared an office with Greg Landrum and Peter Ertl. There are all these amazing informatics folks that come out of Novartis. And I don't mean that to not talk about all the other things, but I want to save some time for talking about things beyond this. But just a little bit about how the international Novartis ended up having all those innovative informatics capabilities, since you were the CIO there, might be interesting. There it was the opposite. You were the management and you had to nurture everybody else.
Listen, Barry, Novartis it's a powerhouse. I mean, it's a research powerhouse. There's no place like it, in my opinion. At least when I was there, it's loaded with talent, loaded. Okay, and it's talent at scale. So they have a lot of great people, a lot of good ideas. It's a large, complex organization, of course, right? But the talent is unreal.
Great. Do you want to say anything about Arsenal and what you're doing now, or should we move on to Chris?
So exactly. So now my career is reasonably broad, actually, and every different role has been a school to me, has been like a university. For example, before I go to Arsenal, when I left biotech and pharma to go to a CRO, Covance, right, to build a technology platform in clinical trials, that was an incredibly different environment. In fact, I'll make a joke because I just made it to the person I talked earlier today, if I have like a couple of minutes. Somebody asked me, what is the difference between pharma R&D and a CRO? And I remember giving the answer spontaneously. I said, in pharma R&D, our job is to spend money. In a CRO, the job is to earn it. that clarifies your thinking about priorities, to a great degree.
You can see Chris and I shaking our heads as people who have to earn our money. So
it gives you focus. And now, I turned 60. Don't look at my youthful appearance, but I am 60 years old, 61 to be exact. And I was working with a private equity firm for a long time as an advisor, and they asked me, last year, a little before them, to join them full time as a partner to help them with the digital data and analytics. Arsenal is a very well-known private equity firm, very successful. It's about $11 billion under management. And you might know some of our portfolio companies, Certara, WCG in clinical trials, Cellcarta in biomarkers. We recently acquired Endpoint, an e-clinical platform, market access from Fortrea or Old Covens. Anyway, it's a great environment, and I'm learning a lot because I see now the world not just from the biopharma perspective, not just from the vendor's perspective, the CRO perspective, but now from the investor's perspective. And that is incredibly valuable. So for any of the audience who are worried about depth versus breadth, I can tell you that every one of these experiences has enriched me immensely.
Thank you for sharing those behind the scenes stories. Chris, I was just, for some reason or another, I was thinking of Bill DeGrotto. I don't know if you know who he is, but he was at DuPont doing kind of like academic work on de novo protein design, which is the most recent Nobel Prize. When he was in industry, and then in academia, he's doing, industrial type drug discovery work. And so I'm thinking back to your background at University of North Carolina and some of the things you've done with academic professors within industry. And our collaboration, publicly announced collaboration at Pfizer is one thing which I'm very proud of, as well as your having co-founded the Pistoia Alliance in Pistoia, Italy, of course, and our work with you with the Gates Foundation with neglected disease, while at Merck is something else publicly announced. But maybe just share a little bit about some of the things you were doing with Pfizer, with us, and with other technologies, and then now you're seeing it from both sides at EPAM, which you also had a hand in, I know, but anyway.
Yeah. So likewise, so thank you for the invitation to actually come and talk today. I appreciate it. I'm honored to be on the stage with my friend Demetrius. This is excellent. Also, I have joined the 60-year-old club, although unlike Demetrius, I'm in complete denial and continue to act like I'm 27. If you don't believe that, ask my wife. Yeah. So I've been blessed to have a really fun career. I've done a lot of different things. I started in government. I've kept a foot in academia all along, or at least tried to, working with Alex and the guys at UNC. I spent the vast majority of my career, as 11 years at Pfizer, doing a variety of things, and had started there doing computational chemistry, moved into chemistry informatics when you and I started doing some work together, and the kinds of things that we were doing with my team there and building out the infrastructure to support all the modeling and the cheminformatics and ultimately the bioinformatics and predictive modeling across the organization. It was kind of like Novartis. It was an incredibly innovative place. While I was there, I enjoyed it. It's a powerhouse again of capabilities. These large pharmaceutical companies have a lot of resources to do a lot of really amazing things, and I'm very proud of the work that we were able to accomplish there. Even what's interesting about the things that we did, we made decisions that were, I thought at the time, pretty um, pretty advanced, right? Because we were adopting and employing service-oriented architecture, microservices, whatever, long before people really started thinking about it, right? And then the kinds of work that we did to provide best of breed capabilities in these hybrid ecosystems, I think was interesting and revolutionary. And now it's just commonplace, right? Every day I get a press release about somebody that's integrated a new NIM or something into some sort of ELN somewhere. So I think it was foreshadowing an ecosystem that is flourishing today with people being able to deliver best of breed capabilities in these technologies that are powering really clever and innovative pipelines. You mentioned EPAM. EPAM's an interesting experiment for me. When I left Merck, I tell people I came to EPAM for a 1-year experiment, and that was 7.5 years ago. So the experiment is either going really, really well or really, really poorly. But either way, I'm still here and having a really a lot of fun, and I'll get to see a lot of what my friends in the industry are doing. I work, and my teams work for pretty much every large pharmaceutical company on the planet, and companies such as yours, right, to empower these large companies to do things. And we're very, very pleased with the partnership that we have with CDD Vault and our engineering to support the vision that you have. So, and that, what you've got on the screen now is I think a world-class solution for how to create, render, modality agnostic stuff on the desktop, right? So yeah, so there's where we are.
Yeah, and we've embraced the open source catcher and really appreciate advancing the chemoinformatics with you and with Greg Landrum for RDKit, with new capabilities such as like atrope isomers, very technically detailed things to sort of push the envelope, macromolecules. We just had release notes with cyclic peptides where they look right from the chemist perspective, even though all the bonds are being stretched because of the nature of the constraints on entropy. So this is one example of a collaboration with you. And the other one that I wanted to mention, and similar to what Demetra says, the way we got to talk about this was by publishing it. And then once it's published, it's, something to talk about. I don't know. I mean, the things that I would say is this after Pfizer had bought Pharmacy Upjohn and Park Davis. So it was probably the largest ADME-Tox data set in the world, all done under standard conditions. So we kind of used the most important valuable data to compare different models. But maybe, yeah, if you want to share anything about this, or I can.
Well, I mean, so I mean, just sort of philosophically, I've been a big fan of open source solutions for a while. And while we were at Pfizer, and this is one of the experiments that we did, I mean, we were constantly keeping track of what was going on in the open source relative to the commercial products that were out there, which were, expensive at the time and still are. And a lot of that exploratory work and got published. This is obviously an example of open source descriptors versus things that you needed to buy from someone. We did the same thing in the cartridge space. We do the same thing in the drawing tool space. And I think open source is interesting, right? In the fact that some of the tools are as good as the commercial tools. Some of the tools put pressure on the commercial tools, and I think a lot of that comes out in the Pastoria Alliance stuff that you mentioned earlier too, right? The idea that there's a lot of capabilities that we have that are in the open source that are necessarily or should be just declared pre-competitive and just put in the open source, and then we just move on, and then we take the resources that we have in terms of human capital and otherwise and devote it toward doing more innovative things on and around those open source pre-competitive solutions that are out there. And this was just an example of, can we actually build something meaningful using open source? And in fact we did. So I was very excited about this research, and it's good to see Rishi's name on there. Rishi, speaking of Novartis, it comes around, right? So now Rishi's at Novartis. So
And
cool.
Eric Gifford's at CDD, so I just thought I'd give a
Exactly.
shout out for him as well. He was with you at Merck. So
Yep.
going back to the Greek philosophers, Demetris, this is a nice slide or a hat tip to you. The reason I know you had like half a dozen of these great quotes that I like this is there's something very pure about the science and the informatics, much like software, in that where the atoms are, their charges, their vibrations, and their rotations and such affect what they do. And if you get it right, it does the right thing. And if you get it wrong, it does the wrong thing. So I just felt this was a very interesting quote. Not sure how come you picked it from your perspective, but I thought I'd make this as our segue from introductions to talking about, chemoinformatics, bioinformatics, chemistry, biology. So going back to the early Greek philosophers, I don't know if you had any comment on this. Otherwise, I'll ask a regular question.
Oh, the only comment is and this is my advice to the audience, guys. You are hearing opinions, an N of 1, N of 2, and N of 3. Okay, it's a very small sample, and because I find myself at this advancing age, increasingly eager to advise people Make sure you synthesize opinions, because in most cases in life, the control experiment is not done. There is no control experiment. You make decisions to the best of your knowledge and your ability and intent. You try to make them work out. That was the intent of this. That don't forget that there's a lot of different opinions in pretty much everything we do, and every one of them is valuable.
Well, thank you for setting the philosophical tone. I could spend the whole time talking about philosophy. I'm going to go back to the original topic of today's webinar is drug discovery informatics for big pharma. And I do think, you know, opinion or otherwise, you're very much experts in the field from hard-earned experience. And for me, the question is sort of a market question. What do you see as the same? What's different when you're looking at a big pharma versus a nimble biotech for informatics? And yeah, how would you look at that? What's conserved? What's the delta?
Chris, you want to take this first?
I can start, and we'll riff on it a little bit. I mean, it's an exciting time, right? I mean, even since we prepped for this call, technology is the bar's moved again, right? So, and there's nothing that's more overhyped right now than generative AI, right? So everything that's going on right now, almost every big pharma and every small pharma has some sort of a project going on related to generative AI. Most of it is POC work, not a lot of it is in hardcore production at this point. And I think that's part of what we have to do going forward is move a bunch of that stuff into production. I think the difference between what I'm seeing in the biotechs and in the tech bios and in the large pharma is that the biotechs that I work with, or the tech bios specifically I work with, are moving rapidly to invest in these and build new technologies to drive these novel ways of doing drug discovery or novel ways of doing or powering pipelines. And it's kind of like the old Google model. They're constantly in a beta mode, right? So they're using these tools in anger to get stuff done. The big pharmas are making investments, but what I'm seeing is a lot of POC and a lot of POC-itis, you want to call it that, and just trying to figure out. And I'm speaking specifically in the early discovery space. There's stuff going on way downstream in commercial and other places where it's even a little more challenging, right? Because it's more regulated and you got to be careful what you do with those technologies. But I will say that the things that I'm seeing that are similar across the two are still investments being made in data and data platforms, and people sort of a renewed interest and awareness that we need to get our data under control, data collection, data management, data metadata, whatever, so that we can generate data products that are analytics ready and Gen AI ready or whatever ready. So I'm seeing commonalities in that awareness and that investment level. And then, like I say, I'm seeing sort of differential investments in the POCs, the MVPs, and the pre-production kind of stuff that we're doing in the Gen AI spaces. I don't know what you're seeing differently, Demetrius.
No, I agree, Chris. I would use the POC. I would completely agree. I call it pilotitis, essentially.
Yeah.
But if you want to distinguish the two environments, the biotech and the big pharma, again, in my experience, and I want to impress this upon your audience, okay, it's just my experience, is that biotechs have the advantage of being very singularly focused, very focused. At 3DP, our vision was to create this engine, right? The mine combinatorial libraries in a virtual way, robotically synthesize the molecules, measure them in bioassays, formulate, improve structure activity models, machine learning models that would correlate chemical structure with biological activity, and use it to keep fishing into the combinatorial space for improved molecules. Okay, but it was singularly focused on this. And fast forward 25 years later, I find myself at Generate Medicines, the flagship unicorn in generative AI for protein design. And it's again the same idea. But they're singularly focused on this. The whole company is built around perfecting that idea. In big pharma, I find big pharma to be more vulnerable to fads. I can use this, things that come and go and come and go. And there's a little bit of attention deficit. There's too many, by the very nature, the model is such that they source ideas from all over the world, essentially, the entire ecosystem. And naturally, which is a great advantage because you experiment with a lot more things, you can try a lot more things. But they're generally less agile for understandable reasons, okay? Diversity, to use our familiar. A bigger company has more people, has more experience, valuable experience, but more diverse experience. And you need to synthesize all of these opinions and views to come up with a strategy for your company. It is naturally more complex and more slow, okay? So that to me is what I've experienced as the biggest difference between biotech and pharma. On the other side, of course, pharma has a lot more resources. Okay, you can do a lot more things with it, but I think there also lives an opportunity or room to abuse these resources and not use them with the most, towards the most valuable problems or the most valuable outcomes.
Mm-hmm.
Yeah, that's what I would say. I think that's the difference between Chris.
I was just wondering, Demetrius, as you were speaking, I mean, I'm thinking about like when I was at Merck and and Pfizer before that. I mean, we made huge investments in human capital, right? So we had a lot of people a lot of smart people that did a lot of wonderful things. And there were investments that were made in technology. And I'm just thinking about the tech bios and the biotechs. If you think about the investments that you make in people versus the investments that you make in technologies, do you see the balance being different across pharma and biotech, tech bio? It feels like the technology investments are larger on the tech bio side, maybe, or are you seeing that differently?
No, I don't think so.
Okay.
Again, I think it's the focus, Chris. But I can tell
Okay.
you the top biotechs, even the startups, have the ability to recruit exceptional people. Present company excluded, of course. The ability to recruit outstanding. Because that's where the future is. That's where the latest and greatest innovations typically come from.
Yeah, one of the things you mentioned early, Chris, was, the AI being as good or as bad as the data and the data consistency. And that's something from day one that we focused and obsessed on with CDD. Our first module was just the structured registration system, activity and registration for entities and their bioactivity. And it is interesting to see that the AI wave coming at the NVIDIA GTC conference this week I was at, and they also were talking about, how the foundation is having the right data model for, your bespoke problems. I did, while you were talking before sharing too much about CDD Vault, I wanted to tip the hat to ABCD. And just, you can kind of see here, even from just earlier about some of the publications, if you want to read up on it, and screenshots, just how, beautiful and the breadth and scope of the technologies there. And this is somewhat rare. I know like Helm came out of Pfizer and is a Pistoia Alliance and some other technologies that you've made available for the world. And so I guess, how do you see that challenge changing with AI for drug discovery and with big
Can you go
pharmas?
back to this? Can you go back to the previous slide, Barry,
Sure.
if you may? Thank you. So let me offer my own perspective, guys, and I think it's the singular most important factor for my career. To me, the secret. I'll paraphrase James Carville, who's Bill Clinton's campaign advisor. He had coined the famous term, "It's the economy, stupid."
Right.
Okay. I've paraphrased this. It's the ops, stupid. And maybe because of the way I'm wired, because I'm such a visual and have a very keen taste for aesthetics and things. I had this instinctive understanding that at the end of the day, what matters is what tools you put in front of the masses, not the experts, not like a handful of people, data scientists, who can work on a problem, but can you democratize data and models? And ABCD was just that. It was a democratization of all the beautiful data that had, J&J had collected for decades, essentially, but brought them in the hands of quote unquote common mortals, the chemist and biologist. And there were two aspects to this. The first aspect, which you see on the right hand side, is to create, and that has direct analogies actually with the present day, to create a structured, organized repository where this data would be available at the speed of light in an analysis ready format. And pay attention to my words. They were not, I don't use them. It's not, I don't use superlatives randomly. At the speed of light. Let me give you one anecdotal example, Barry. When I built single-handedly with a colleague of mine, the substructure search cartridge for ABCD, okay? And we had 10,000 queries posted, historical queries that J&J researchers had posted on their historical databases that we used to assess the algorithm, validate the algorithm. And I remember making a claim to the team that I'm going to get every single one of these 10,000 queries operating on a 5 million member library at the time. I can't even remember what the size was, in sub-second timeframes. I deploy the first release, and the slowest query is about 2.5 seconds. And everybody says, we're done. And I said, no, we're not done. We're not releasing this until every single freaking query will run in sub-second time frame. A week later, every single one of them was sub-second. So it is, so that's the back end. And I contrast this today with the data lake paradigm, where people put data close to its source format into a repository, and somehow this data is now fair. It's findable, yes. It's accessible, yes. It's interoperable, yes. It's reusable, maybe, but it's not usable. So the thing is to take data and bring it to life, right? Make it come to life in the language that the user understands. And the same, parenthetically, applies to models. And I think one of the most important decisions we can make as a community here, or people who deal with those systems, is to really think hard about how you democratize and operationalize AI and ML models, okay? And think about people who are really good at this, and these people who are really good at this are people who work on consumer types of businesses. Netflix has very sophisticated models on the back end, yet the user experiences as recommendations in their workflow, embedded in the natural workflows. So that's the moral of the story, right? Make data accessible instantly in an analysis-ready format. An analysis could be computational or visual, and bring tools that, typically every one of my presentations end with a reference to Vitruvius, a Roman architect, studied ancient Greek, early Roman architecture, and distilled the essential elements of every great structure into three things: firmitas, utilitas, venustas. Strength, function, beauty. So my career was about building systems that were powerful, functional and intuitive, and jaw-dropping gorgeous.
Well, I agree. I want to riff off two things you said, and then I've got to give the floor to Janice. Otherwise, we'll just do the whole time without going into the technology. But the first thing was making the technology democratized for the masses. We've always made this for regular experimental scientists. Our tagline is complexity simplified. It's in my background here. And that whole idea of making it that, just like surfing the web or something, any chemist or biologist can get at the data the way that makes sense to them is a biggie. The other thing that, and again, when I saw your technology was so far ahead of the times, with the new AI and the deep learning and such, we've looked at where is it different and really practical. And I remember a benchmarking between two commercial cartridges talking about the minutes it takes to do a substructure or similarity search with an indole for some reason was hard to encode. And one of the things that Peter Geddick's done, and because he was at Novartis, he was thinking about this for 20 years, and we've been working on it before OpenAI. So we've encoded all the molecules as chemically rich vectors, which means that it's super rapid to do similarity search, unlike the previous technologies. So within milliseconds, we can go out milliseconds to millions of molecules from Chembl, Sure-Chembl, enamine, representing billions of molecules, and do that with the speed of previous generation technologies. So even if like QSAR models are all about the same, like we showed back at Pfizer, and it depends upon the data and democratizing the data and models, there are some real advances. And the other one that we did around the AI module is to make the generative bioisosteres chemically aware. So it would be constrained by the real molecules in Chembl and not suggest things that are like 8 9-membered rings that won't cyclize because of the bond angles, won't have Payne's filters, microacceptors, things that will cause false hits. So we could constrain these so that it makes sense for a med chemist intuition for the AI capability. But at the core, it's about making it easy for scientists to have a whole solution for drug discovery informatics. And so I wanted to show the ABCD slides to sort of set the stage for this. But I'd love to show you some of the technology and get your impressions from Janice, if that's okay.
Sure.
All right.
Yeah, go ahead, Janice. Yeah, she also did biology screening at Merck and BMS and biotechs before she came to CDD. But yeah, go ahead, Janice.
Yep, and I used to be a customer Vault administrator myself, so I am going to show you the technology. As you know, we work in a multimodal world, and this is just an example from my own private secure system. So my vault of data that is stored in here to kind of show you examples of what our customers typically store in their own private secure vault. We are a registration system. With that registration system, you can give us file string, mol file text. We are now registering your compounds uniquely. We are always going to perform this duplicate check. We do auto calculate some physiochem properties. You can store aliases for those things that you're registering. So, you talk about smart people that do wonderful things. We've been around for 20 plus years, and we have smart people behind the scenes doing all these wonderful things to create the foundation for you with the system fields. So like you can have a synonym here to capture, in essence, what that entity is that you're registering. And typically and traditionally, we've been about the small molecule. You get auto-generated breadcrumbs to electronic laboratory notebook entries. Again, just here now auto-generated for you as a system field. We have an inventory system because with that registration system, you want to keep track of what you have on hand and where you have those things on hand. On top of that foundation of the system fields that you get, you can create your own user-defined fields to capture attributes about the things that you're registering. The data is only as good as the data you put in. Here, if you store a file, as one of the attributes for the thing that you are registering, you can mine that out of the database. So again, this is your central repository, and we work through collaboration. We work in a multimodal world. Biologists and chemists work together. This is intended to be a single source of truth. And here, connected with those things that we register, the assets that we register into our vault, we have our experimental data, so protocol information. We store data in what we call protocol definitions, or simply protocols. It's how are we going to store the data, what we're going to do with that data. We might want to plot this data. In plotting this data here now, I have access to flag outliers and override. When I click on that, I get to this screen where I can hover over a data point and actually mark it as an outlier. And as soon as I do that, here I'm using the 4-parameter logistical curve fit. to fit this data. As soon as I do that and I go back into the system, you can see that this value, unlike the other values here that are displayed in plain text, this is underlined and italicized, telling me that, there's something different about this, and it's because I flagged that outlier. So this value is calculated excluding outliers. So from beginning to end, from the researcher that puts in the data to, the high level user who's going to come in and mine that data, we know what's going on. We can make calculations, we can average then those values. And if you then look in here, we can store any type of data, including PK data. We can, with our Curves add-on, actually analyze this data in different ways. We talk about setting the bar high and the bar keeps moving. Traditionally, we've been about the small molecule. We actually have different registration types to register your antibody therapeutics. We have gone into the macromolecule. A macromolecule composer is also offered. And here we're using the SESR model, and you can actually get that full atom view of a cyclic peptide that's now registered in the vault. And then I'll continue on just to show you the different types of registration types. You get the plasmid map, and instead of the small molecule, you've got the letter block sequence for a nucleotide. We even have the ability to capture formulations. Sitting on top of this, you see the data in rows and columns of data, but we have an unstructured space. So a place to capture your experimental write-ups, your synthesis. A chemist can come in here, write out their syntheses, even register compounds directly from the electronic laboratory notebook entry. You have now data write-ups for the biologists who can come in, lay out their experiments, go through and stepwise through their protocol instructions, capture other information that wouldn't necessarily go into those rows and columns of, database tables. And at the end of the day, this too is searchable. So we search for our terms and we find the connected entries to those things. I'm just going to step in here really quickly to visualization. Now that we've put our data into the system, we've registered our compounds. We have experimental data for this as well that's displayed here. We can launch into visualization, and launching into visualization, you get an XY scatter plot at the top of the page. By default, you've got your data that's pushed into the data table down below, histogram filters on the right to work with these. You have the ability to now visualize the data, compare your molecules, your assets that you're capturing data for in your vault to best identify what your leads are. You can add histogram plots, 3D scatter plots, perform an R group decomposition, cluster analysis. At the end of the day, what you're looking at are things like this to now visualize our data with that R group decomposition. We can see, what data we've supplied, what we have available, what we've made, what we didn't make. Cluster analysis will then allow us to perform a clustering of now comparing our molecules against one another and fitting them into a cluster identification based on a threshold score. And now I'm looking at the distribution of those different molecules that I make. Ideally, what we'd like to do, we have a system, and while this may seem simple, it's the ability to mine that data, simplify our needs so that we can easily pull together, a presentation. Here's our data. Let's favorite some compounds, which I've done here, and let's push this data. Now I've set up a data comparison table, and let's look through the data, and we can see this data in different ways. We do this through saved searches. We can, save our visualization. We can save the searches themselves so we can execute those searches again and again for important data that we're mining out from the system. That's what I want to say and introduce you. It's a very high level overview. If you want to take the time to learn more about this, and we'll have a poll at the end for you to, weigh in on. But yeah, back to Barry.
Yeah, why don't we run the poll now for people who want to see more? I don't know if you have that, Joshua. Yeah. So, yes, I could vote myself too. But so, I'll give you 3, 2, 1. Okay. So, it was sort of a whirlwind overview, but basically a whole solution for drug discovery informatics today, while still doing it complexity simplified. So this is the quality of the software with our engineers, and the design is, 10,000 automated tests, 4 to 5 lines of QA code for 1 line of production code. We're building this for perpetuity, like a LinkedIn or Facebook. And so it's taken a lot longer to get all those capabilities, but once we do it, it's good and reliable with like 99.99% uptime, 7 million logins, 4 billion data points. It's probably the most drug discovery data in one database in the world. 20,000 research projects. We're over 690 companies and academics using this. So I guess one of the things, if you're in my shoes, we can lean into the AI capabilities for the shiny new and differentiating capabilities, or focus on more and more of the whole solution, the other 8 modules. So there's sort of, I guess, 2 questions. One is for leaning into the AI capabilities, which I know you shared this slide here, Dimitris. And I can tell you from going to the NVIDIA GTC and seeing them at JP Morgan that the world is investing on this outcome. And it's coming just because they're exponential curves on top of exponential curves now. And this is some very interesting things that they're doing and setting up with, and they announced our logo on their slide earlier at GTC. The things that we've done is not to say this is a hole-in-one, a drug for something as complex and multi-parameter as that, but to humbly give some suggestions for new molecules, and doing this conservatively so that it suggests ones that are reasonable, are not going to be microacceptors, are not going to be, have pains, or are going to be things that are synthesizable for the generative chemistry. And the other interesting thing is you can do this iteratively because of the catcher sketcher. So we can take any final molecule that you're screening for a drug candidate, erase half of it in catcher sketcher, run the bioisosteres on the starting materials for what you would want to make. Any one that you could buy off the shelf from Enamine Real Library, you can just click and buy it. And if you want to see if your molecule's been patented or not, you can go to the 24 million from SureChemble, which is SureChemble are the publicly available databases for novelty of your bioisosteres. So it doesn't take away the human medchem intelligence. You still look at it, but it democratizes it, right? So you get all sorts of ideas for new ones, and they're reasonable. And you can look at whichever fragment you want of the molecule to make bioisosteres generatively. Then for either your parent molecules in the database or others, if you want ones with higher log D or lower TPSA or CNS MPO, it's automatically calculated. And you can go into the similarity of these millions representing billions of other molecules. And this happens automatically within milliseconds for everything registered, or anything your imagination can draw in your wonderful open source sketcher here in the bottom. So, we're a few years ahead of others because we were working on this with SBIRs back in 2018 for the deep learning. And feel we have something that's state of the art in the chemistry space. And of course, biology is based on chemistry, right? So we have the ability to register on natural amino acids and oligonucleotides for cyclic peptides. You can make bioisosteres off the side of those things too, right? And you can do similarity, and all the sort of exciting things happening in generative biology. You could do that at the atomistic level, and with the chemically rich vectors, you can look in lots and lots of zeros and ones underpinning that information with, the speed, as you mentioned, of a Netflix or a facial recognition algorithm. So, I'm excited about this intellectually because I know it's practical and ahead of the curve, and the space is moving so fast here. So that's been an area we've been really lucky from your groups at Novartis to have Peter Geddick, and working with others like Greg Landrum in the open source space and EPAM. And so we can lean in there. Um, but there's
So Barry, can I ask a
infinite
couple
work
of
to do for
questions?
any of the other modules as well. Yeah, go ahead.
Yeah, just an observation. I guess, uh, as I mentioned earlier, and so yeah, stay on that screen. That's a good one. There was every day there's a press release about somebody integrating a new capability of somebody into their software, and today's was around Sapio integrating NVIDIA BioNeMo into their
Mhm.
LIMS platform, right? So, what kinds of integrations do you foreshadow like that with CDD? Do you see tying into some of those additional capabilities and advanced capabilities provided by others? What are you thinking in that space?
Yeah, that's a great question. I do. I think the thing that I want to mention is, you saw that the front end for these things called human beings and people from Janus, that it's all very intuitive and a complete whole solution there for your private data and secure collaborative. Um, there are two areas that we were ahead of the curve. I mentioned before 2018, before the sort of deep learning AI explosion with OpenAI, ChatGPT. Um, but the other thing is we were hosted before AWS existed when we spun out of
Right.
Lilly in 2004. So that was a long time. And we got all the arrows in the
Mhm.
back when no one would trust a hosted system, and all the arrows in the back when someone takes a behind firewall version and throws it up on the web and says it's a web first application, which it's not. So this was built for internet collaboration, first with people, but second with algorithms, with software. So if you can do a little bit of command line with our automation module, which is a kind of fancy word for API, we can do gets and puts. So anything in CDD Vault, you can do a cron job at 3 a.m. and pull out all the data or your most recent data into your other bespoke application and do your modeling and bring it back if you have your own favorite, flavor of the month software. So we integrate with Schrodinger, with Certari, you mentioned, Demetrius, you're an investor in, with Optibrium, Tijan, Microsoft's inside our ELN. So, we'll make smart build or buy decisions, whether collaborating with you folks at EPAM or, working on our own. When our own's best in the world, we'll use it. When someone else's best in the world, we'll integrate with it. And so one way to do that is with the API. Another way to do it is, with native tools, code within it, within the natural workflows. So that's how
Just
we've sort of done that. Yeah.
no, it's excellent. And just a real quick follow-up question. Just also thinking about the things that we're working on these days and the clients are asking us to do related to democratization of access to complex tools, right? So everybody loves agentic workflows now. Do you see an opportunity to bring some sort of agentic workflow layer to CDD and actually allow people to have a dialogue with their data, right, or reason with their data in a workflow driven way using agents to give them access to the capabilities and services that they need in real time?
That's a very interesting question, and like crystal ball predict the future type question a
Okay,
little bit as
okay.
well. There's so many jokes I could tell, but anyway, what I will say about it, moving outside of our space, there's been an interesting dynamic. You can see it on LinkedIn, like the CEO of Box.com talking about his bespoke software application or Salesforce SaaS businesses versus the larger companies like a Microsoft talking about the agents world. So I will say there is a battle in the marketplace between applications and agents. Let's just say that
Right.
in the horizontal marketplace. And so that's going to play out. We've sort of done the Salesforce model to SAP, where you do the 80% that everyone wants, what Demetrius was talking about for the regular experimentalist, not the specialized thing for someone like a Rishi or Eric Gifford, who really, knows the modeling. And that's been an excellent strategy because that's 90% of the scientists. I think it's really interesting to think about with AI how much it helps those who are most sophisticated and most expert to get better and level up their game, and how much it helps people who are maybe not as expert. Maybe they, maybe they're perfectly smart, but they're not like, focused on that area. They have a different level of expertise, but guess what? The technology can help them level up. And so, whether in
Yeah.
sales or marketing or science, all of this is coming. The wave's coming, and it's going to be bigger and more than you expect, right? And it's the nature of like the future shock book, but basically you have exponential curves on top of exponential curves. We've had exponential logins. NVIDIA's having exponential revenues now, and there's a whole ecosystem. So I guess I'll say, whoever doesn't have it, once you can imagine and ask a question like that, it's going to be happening. And to me, it's kind of going too slow because I'm always like thinking faster as to what's going to be there. But just the nature of progress, right? We had the printing press. Now we have the internet with information for everybody. There's going to be a bit of an explosion there. I still think the internet was the biggest revolution of our time. Maybe I'm getting older too, because it brought all the brains and all the people together, which is the foundation for the AI training set. I also think it depends on the problem. So LLMs have the training set for language, for biology, and for understanding the language of life. There was a recent publication with Evo 2. I don't know if you saw that from the Arc Institute, where they looked at 9 trillion base pairs, and they took it from all these different species, just one from a human, and they're able to see the human mutations for cancer from, first principles, basically, and design new organisms such. So, we're going to have to put in our seatbelts and see, because it's evolving on all fronts simultaneously.
Ari, can I add the thought to this a little bit? Is that okay?
Yeah, yes, spasiba.
First of all, first is a comment, not a point. I'm actually envious of you that you were able to build a business. The software looks great, Barry, and I've built these types of tools before, and may not come as a surprise to you that at some point, because there was great demand for it, I did explore potentially externalizing ABCD and making it a commercial tool. Didn't work for various reasons, but I'm envious. I'm envious of what you've been able to build, because your impact on the industry as a whole is tremendous. Now, let me switch gears a little bit. I think in all of this AI talk, a lot of which involves hype, of course, and we'll come to that in a second. People often miss the most important substrate for AI to work, which is clean, good, reliable data. Okay? And getting clean, good, reliable data in a format that is machine learnable, is understandable by humans through visualizations and other tools, is perceived as drudgery, a little bit more mundane type of work, but it is profoundly important. If you can allow me to make a reference here,
Yeah.
I'm not sure if you know this, but one of the most recent investments that we made at Arsenal since I joined is to acquire Rancho Biosciences, which is, you probably agree with me, the world's leading biomedical data curation and increasingly data analysis company. And why is this? Because we believe that this is the substrate, having clean, well-organized, standardized, canonicalized data is foundational to building reliable, robust models. And parenthetically, so
yeah, just we have 1 minute left. I'm just going to say
we do?
one thing or two things, and then we'll see if we have a few time for a question or two. Doesn't surprise me, but my last company, Libraria, built the first gene family-wide SAR databases and combinatorial reaction databases. So I'm familiar with the value of well-curated data, and with CDD Vault, and it was copied by like half a dozen companies very quickly that were more successful than myself. So I made all my mistakes on that company and learned my hard lessons. But If you look at what we've done here to build a barrier to entry, a real moat is we capture people's own proprietary data that becomes their composition matter patent, their bioactivity patent with activity and registration. The assays module lets you capture metadata around the assays. So you can do chemoinformatics, bioinformatics, assay informatics, and then the curves for the different types. So completely agree with you that the quality of the data, the curation matters. The models are only as good as the data. And that's sort of been our focus. It's just with the AI and the generative bioisosteres and the ultra-fast similarity search, we had something very novel. And so we've kept some of those capabilities trade secrets and made that a module where we leaned in. But, there's others. The Nobel Prize with AlphaFold 2 and David Baker from UW was for generating protein structure from sequences. So I think there are some real advances. There was a CASP competition where, for years they've been sort of incremental progress, and then there was a big progress. So it is as good or as bad as the data. The nice thing about that competition is everyone had the same data, right? They had the sequence of the protein without the structure. So it was a legitimate experiment. But drug discovery is hard, and chemistry and biology is hard to predict things, and it's multi-parameter. You're doing a model of a model of a model, right? So you're looking at a small molecule against a protein to model its activity in a cell, to model its activity in an animal, to model its activity in people, in clinical trials. So it's still very much an empirical science where you have to experimentally test things. And your data is not perfect for getting how it's going to work, but it will keep getting better and better, and it'll get better, like you said, as a function of the quality of the data and how general or how specific each training set is, sort of the scope and limitations. So I come at this AI area. My first paper was in combinatorial chemistry, which is to experimentally test things, looking at everything without any models or predictions, if you will. And so very look at all these things. And I think, as you both know, having created technologies, when you create them yourselves, the scope and limitations of it very, very well. You know where it works well, where it doesn't do anything at all. And so, our mantra has been focus on doing the very, very simple thing of getting your data correctly. All the things you saw from Janice for the different types of curves and structures, we can handle the chemistry and biology correctly and the bioactivity correctly. And then each scientist and their own intelligence and their own models or others, it's going to determine how well they do that game. I am sensitive that we, of course, are out of time. I'm not surprised at all. But I guess I leave it to Chris and Josh if there's anything you guys want to wrap up. I want to be sensitive to folks' time. We still have over 100 people still hanging out listening, but yeah. Is there anything else, Charlie or Josh, to take us home? We can answer the questions by email if folks send us an email to info@collaborativedrug.com.
Yeah. So if you have any last minute questions, please get them into the Q&A tab. We will respond to them via email. Sorry we didn't have a chance to get to those today.
Okay.
But thank you for everyone for your time and attention. Thank you, Demetrius, Chris, and Barry as well. It was a fantastic discussion. That was very, very fantastic.
Thank you, Chris and Demetrius. Happy to follow up by email with you guys. Thank you for your time and your brains and your careers. You've done stuff worth mentioning, remarkable, and really, really honored to have you here at this sort of pivotal moment where CDD is taking off exponentially and has scientists all over the world using it every day. I think we rest on the shoulders of giants and very specifically your two life's work, both Demetrius with ABCD and Chris, what we're building now for the future. And this won't be our last time talking. So thank you so much. And that's it.
Congratulations. Thank you.